Welcome to Let's Get to the Marks. We're looking at AS Biology Paper 1. This is the 2023 paper, so let's get into it. Don't forget to like and subscribe. Let's go. So, question 1 is 1.1, and it is about the primary structure of all proteins. Describe the primary structure of all proteins. What comes to your mind? Remember to pause the video, then never go. But what comes to your mind when we talk about the primary structure of all proteins? It's the sequence of amino acids or the specific order of amino acids in a polypeptide. And what type of bonding do you find in polypeptides between the amino acids? That's right, you find peptide bonds. So, with the primary structure, you need to know about peptide bonds. If you're talking about the secondary structure, then it also has the peptide bonds, but we have additional hydrogen bonds. And then the tertiary structure, we introduce ionic bonds, disulfide bridges, and hydrophobic and hydrophilic interactions. So, you need to revise and know the levels of bonding in the 4 different protein structures. Okay. This question, 1.2, is about the genetic code. Define the terms universal, non-overlapping, and degenerate. So again, pause the video, have a go. Now think about the word universal. What does that mean? That means like everywhere, everything. So what is it that everything in the universe, in the known universe, i.e. on Earth, what is it that every living thing has and shares? And the answer is the triplet code. So the triplet code is universal. This means that if you have any 3 triplet of bases codes for the exact same amino acid, regardless of what organism you find it in. So it could be a fungi, a bacteria, a plant, an animal, a protist. You will find those same 3 base sequences on the triplet code will code for the exact same amino acid. So let's put our answer together. This means the same triplet code or codon, which is another name for a triplet code, codes for the same amino acids in all organisms, or you can say living things. Okay. And let's just give a little example. Now, you don't have to know the codons for any specific amino acid, but here's one for you. GCA codes for alanine in all living organisms. Doesn't matter whether you're bacteria, fungi, or human. GCA will always code for alanine, alanine, however you pronounce it. Right. What does non-overlapping mean? This means there is no overlap between codons or triplet codes. And I'll give you a little example, but you would get the mark for just saying that. No overlap between the triplet codes. One triplet code's bases cannot be combined with the bases from another triplet code to code for an amino acid. So each codon is a separate 3 bases, and the bases do not overlap between the codons. So for example, here you can see I've got one codon which reads GCA, that's the one for alanine, and then I have another one, GGA. And what it means is they have to be read separate in 3s like that. You can't take the A from the first codon and combine it with the 2 Gs from the second. It won't be read as AGG at any point. Cool. And that's just giving you the explanation. But all you need to say is no overlap between codons. Now, what does degenerate mean? Well, this means we only have 20 amino acids, but how many codes do we have? We have 4 to the power of 3 because we have 4 possible bases arranged in 3s. So you go 4 times 4 times 4. There are 64 possible codes, but only 20 amino acids. This tells us that there is more than one codon for each amino acid. Of course, we have stop, start codons and some other ones as well, but it means most of the amino acids have more than one codon. So as an example, we use GCA, GCT, GCC, and GCA. These 3 codes or codons or triplet codes all code for alanine, the same amino acid. And that's what we mean by degenerate. Now let's move on. We're whipping on to question 2. Let's go. Okay, let's get into question 2. Figure 1 shows a transmission TEM electron microscope image of a Streptococcus bacterium. 2.1, describe how the appearance of the area containing DNA in a TEM image of a eukaryotic cell would differ from that shown. So pause the video, have a go. All right, so we really want to talk about how a eukaryote would be different. And there'd be a nucleus. We'd also see a nucleolus. Actually, you're not going to be able to see the nucleolus. No, you might be able to see the nucleolus. Yeah, you could make out the nucleolus. You definitely couldn't make out nuclear pores, so that'd be a wrong answer I put there. Don't put that. You will be able to see the DNA is attached to histones. You'll be able to make that out. And the DNA will be linear in eukaryotic cells. Okay, it won't be in a loop like we saw there. Okay, let's get on to the next question. Very similar question to the last question. Describe one difference between the DNA or structure of DNA in a prokaryotic cell and in a eukaryotic cell. So in a prokaryotic cell, the DNA is going to be in a loop. It won't be attached to histones, which are the proteins that won't have histones. And in a prokaryotic cell, the DNA is not linear. So any of these is going to get us a mark, but we only want to describe one. Now, describing two there is fine, or but we want to say but we want to make sure, like, look, what would happen if I said in a prokaryotic cell the DNA is linear, which it isn't, it's in a loop. Well, that would be a mistake. And as they've said, describe one difference, the list rule would apply. The examiner would look at my correct statement that DNA is not associated with histones, but then they'd take that mark away because I've said something incorrect. So when it says describe one, be really careful that you don't say something incorrect. The times when you need to make more than 1 point is when it doesn't specifically say describe one or two. It doesn't give you a specific number of points to make. Okay, let's look at this next question. Streptococcus bacteria can infect the lungs when air is breathed in and cause lung disease. Describe the mechanism of breathing that causes air to enter the lungs. So pause the video, have a go. All right, well, your diaphragm contracts. You will not get the mark for that unless you say and flattens. Okay, so it goes down. And what this does, combined with the external intercostal muscles, these are the muscles between your ribs that contract and help pull the ribs up and outwards. And what does that do? Well, so your diaphragm's going down or flattening. It doesn't really go down, it flattens when it contracts. And the muscles around your ribs, the external intercostal muscles, they contract and lift your ribs up. So at the same time as the diaphragm flattening and your ribs going up, you're increasing the volume in the thorax. And once you have a bigger volume in the thorax, the pressure within the thorax decreases, the air pressure within the thorax decreases. And in fact, the pressure decreases lower relative to the air pressure outside, it becomes less. And that means you get a flow of air down a pressure gradient from the external outside the body to the internal. And you need 3 of these points really clearly made to get the mark. Just remember that with the diaphragm, you've got to say contracts and flattens. And with the intercostal muscles, you have to specify that it's the external intercostal muscles. Right, let's look at 2.4. Some strains of Streptococcus bacteria are more likely to cause lung disease than other strains. Strains that do not cause lung disease are quickly destroyed by phagocytes. Phagocytes are stimulated when they bind to the murine on Streptococcus bacteria. Remember that murine is also called peptidoglycan. Each strain of Streptococcus bacteria has a capsule, which is a slime layer of different thickness from the others. Suggests how the Streptococcus bacteria with a thick capsule are more likely to survive and so therefore cause lung disease. If they have a thick slime layer, not a thick murine layer, because that's in the cell wall, the capsule's outside the cell wall. So if they have a thicker capsule around the cell wall, it means the phagocytes are unable to bind to the murine that's found on the cell wall. Okay, and this means the phagocytes don't get stimulated, which means less bacteria are going to die or be digested in phagocytosis. This means more bacteria survive. We have a higher rate of binary fission or reproduction, and they produce more toxins, making us ill. Okay, moving on to question 3.1. Figure 2 shows a fatty acid that contains 20 carbon atoms and 4 double bonds. So we can see here the 4 double bonds in the fatty acid. And in a skeletal formula like this, you get a carbon atom at the end of each line, just like this. And the question asks us, on figure 2, draw a box around the R group of the fatty acid. So pause the video and have a go. Okay, let's go through the answer. So R groups are the variable part of the fatty acid chain. R groups on you find R groups on amino acids, you find R groups on different biological or organic molecules, and they're always parts that can vary. So you've got to ask yourself the question, which bit of this fatty acid do you recognize as always being there? Because if you can recognize the bit that's always there, that's not the fatty acid. Or, sorry, that's not the R group. So which bit of the fatty acid do you see in every fatty acid molecule? And hopefully you guessed it right. That bit I circled in pink is known as the carboxylic acid part, the COOH, and that is the same in every fatty acid. They will all have a COOH or a carboxylic acid group at the end of each fatty acid molecule. So the R group is the bit that can vary, and that's this bit I'm highlighting in the pink box. So you should have, to get the marks, highlight that bit and not include the COOH, which I've not done very well. But there we go. So once again, just to remind you, you're going to find that COOH or carboxylic acid group of each fatty acid tail in a triglyceride or a phospholipid. You're going to find those are not part of the R group, so they're always remaining the same. All right, let's crack on and have a look at this question 3. We'll zoom in a little and get closer on this question 3. Remember to pause the video and have a go. Figure 3 here shows a triglyceride. You can see we've got a key telling us that we have a glycerol, 3 ester bonds, 3 fatty acids. Describe 2 differences between the structure of the triglyceride shown in figure 3 and a phospholipid. So this is quite easy. We just want any 2 differences. Well, the triglyceride's got 3 fatty acids. Do you remember how much a phospholipid has? A phospholipid only has 2 fatty acid groups. So there's one difference for you. What about the number of ester bonds? I'm drawing a phospholipid here. You can see a phospholipid only has 2 ester bonds. And for a third difference, a phospholipid has a phosphate group, whereas a triglyceride doesn't. So in this type of question, it's important to let the examiner know which one you're talking about, because you could either argue the case for phospholipids or triglycerides. So here I'm making it really specific and saying triglyceride does not have a phosphate group. Triglyceride, has 3 fatty acids. Phospholipid only has 2. So I'm making it really clear I know which one is which, just in case the examiner thinks you don't know what's the triglyceride and what's the phospholipid. And there we go. That's the end of that question. Hopefully you got 2 marks. Again, here it says describe 2 differences. I've put 3 here. I would suggest in an exam you only put 2, because if you put an incorrect one, you're going to lose one of those marks with the list rule. All right, let's move on to 3.3, the next part of this question. Explain why phospholipids can form a bilayer but triglycerides cannot. Once again, pause the video, have a go. Triglycerides are non-polar, okay? They're hydrophobic. And you think about the fatty acid tails, they're also hydrophobic. But what's the difference with phospholipids? Phospholipids have a phosphate group on the glycerol head, so they have a hydrophilic head. This means that phospholipids are polar molecules, whereas triglycerides are non-polar molecules. Phospholipids have a polar part and a non-polar part, but the dipole on the phospholipid means that they can arrange themselves where the hydrophilic head is attracted to the water. Now we find basically life is based on water. So we have the cytoplasm inside the cell is basically water based, and we have the tissue fluid that surrounds the outside of cells is water based. This having a hydrophilic part is essential to the phospholipid bilayer, because it means that the cell membrane can interact with the outside of the cell, the tissue fluid, and it can interact with the cytoplasm, which again is mainly water based, inside the cell too. If you just had a triglyceride, it's hydrophobic, so it's going to repel water and wouldn't be able to interact with either the internal environment of the cell or the external environment. And that's why the phospholipid heads arrange themselves facing out of the cell and into the cell, and the hydrophobic tails of the phospholipids stay in the middle of the bilayer. Cool. Hopefully that's clear. Not as clear as mud, because that would be no good. And you can go through and give yourself some marks. As I say, it's always good to pause the video at the start of each of these questions. And if you're finding this walkthrough useful, make sure that you check out my other walkthroughs because I've done the 2022 paper, the specimen paper, the 2020 paper, and I've got loads if you're doing chemistry A level as well. So make sure you check out all my other walkthroughs. On like and subscribe, of course, because I'm doing A level too. Let's go. Okay, let's look at 3.4. Figure 4. Where are we? Here we go. Figure 4 shows two fatty acids, A and B. If you check out A, you can see it's saturated. There's no double bonds, and B's got a double bond there where it kinks. So those two fatty acids are slightly different. One's saturated, one's unsaturated. Scientists, make sure you use a highlighter, by the way. Pause the video, have a go, but use a highlighter in these questions. Scientists fed rats a diet with added fish oil for 4 months, obtained samples of red blood cells from the rats starting this diet. And after 4 months on this diet, for each red blood cell sample, they separated the cell surface membranes and they measured the percentage of phospholipids containing each of fatty acids A and B and the fluidity of the membrane. And here we've got the results. The time of the red blood cells, so at 0 months and 4 months is when they sampled. Mean percentage of phospholipids, we can see it's gone down from 19.8 to 11.7. Mean percentage of phospholipids containing the unsaturated one has gone from 1.7 to 9, so has increased. And the mean fluidity of the membrane has also increased with the increase of unsaturated as well. Suggests why the fluidity of the membrane was higher after 4 months. Use the information provided. Well, what do we know? We can see from this table of results that the percentage of saturated phospholipids or phospholipids containing a saturated fatty acid tail with no double bonds, that's gone down from 19.8 to 11.7. And that seems to correlate as you have less saturated fats or lipids, you seem to have a more fluid membrane because it's gone from 31 to 97. So I would say the mean fluidity has increased after 4 months, and this is due to the reduction in saturated fatty acids. And there looks like there's been a fourfold increase in unsaturated fatty acids. So we really want to specify that to get those marks. So the mean fluidity has increased after 4 months, and this is as the mean percentage of fatty acid B, which is the unsaturated one, has increased. Now, what's the explanation? Why do unsaturated fats increase the fluidity of the membrane? What do they have that the saturated fats don't? Do you remember the diagram we looked at? So what the unsaturated fats have is a double bond known as a kink. Wherever you get a double bond, the fatty acid tail bends. If you're a chemist, you know that's because double bonds have no rotation around them. So the atoms either side of the double bond can't rotate and it causes a bend. So the more kinks you have, if you're polyunsaturated with lots of kinks, you're going to have a very low melting point and increased fluidity. It just so happens for those of you that are looking after your health, you should look for sources of fats which have lots of unsaturated fats. Because of their low melting point, they stay fluid in the body more and less likely to cause atheromas and clog up your arteries. Anyway, I've put all the answers here for this one. So let's go up to the diagram and show you what we mean by the kink. Okay, so over here, look at fatty acid B, and you can see here we've got a double bond, right? Now, it's those double bonds that cause a kink. Now, this is only a mono unsaturated fatty acid. It's got one double bond, but you often get lots with polyunsaturated. Think olive oil and things like that. The kinks basically weaken the forces, the intermolecular forces you learn about in GCSE chemistry that exist between molecules. And so they lower the melting point because those kinks stop the fatty acids approaching each other as closely as they'd like. And they weaken the forces between the molecules. So there we go. Cool. All right, time to move on because I'm waffling. Okay, question 4. Scientists investigated the activation of T lymphocytes. The scientists studied two types of cell. You've got naive T cells, which are two cells that have not yet been in contact with a foreign antigen. They're naive. Activated helper T cells, which are T cells that have been activated by a foreign antigen. Figure 5 shows the electron microscope images of the two types of cells. So we got the naive T cell with P to Q on it, 2 micrometers. We've got a little scale there. We've got the activated T cell has a volume of 463 micrometers or micrometers. Calculate the volume of the naive T cell shown in figure 5. Calculate how many times larger the activated T cell volume is compared to the naive T cell volume. Assume the cell is spherical, which means we can use their equation for the volume of a sphere. Use PQ to measure the diameter of the naive T cell. So we're going to get marks for measuring the diameter of the naive T cell and then calculating the volume of the naive T cell and then working out how much bigger the activated one is. We've been told the activated one is 463, so we definitely want an answer smaller than 463 because they've said how many times bigger is the second cell than the first one. And what you want to do here is measure the length of that line. Now, it's going to be, if you've got the paper in front of you, it's going to be different. So I'm going to tell you this is 18 millimeters, but it might not be 18 millimeters. It depends on the printout you've got. Obviously, that affects it. Now, the ratio should be roughly the same. So those 18 millimeters equals 2 micrometers, which means I've got a scale of 1 micrometer is 9 millimeters. Now, I've measured that line as 44 millimeters, the line from P to Q. The 18 millimeters is the line with the 2 micrometers, my scale, which tells me it's 1 micrometer to 9 millimeters. So I'm going to do 44 divided by 9, which means my diameter is 4.8 micrometers. But we need to use the radius. So the radius must be 2.4 micrometers, which is half of that line. So now I'm going to do my calculation, which is 4 divided by 3 times pi times 2.4 cubed, which is the radius. Make sure you bracket everything in your scientific calculator correctly, or else you won't get the correct answer. So remember, I worked out the line was 44 millimeters, P to Q. I used my scale to convert that into micrometers, and then we're using half of that, which is 2.4. So now this gives me 57.90 micrometers cubed. That's the volume of my naive T cell. But how many times bigger is the activated helper T cell? Well, I'm going to round that 57.9 to 58, and then I'm going to do 50. I'm going to do 463 divided by 58. And it sounds right, doesn't it? Because we knew that the second cell must be bigger because they asked us how many times bigger. So 463 divided by 58, I get 7.98. Let's round that up and say it's 8 times bigger. And that gives us our lovely 3 marks. Boom! 4.2, state one feature that shows the images in figure 5 were taken with an electron microscope and not an optical microscope. Optical is your light microscope. You used a lot of GCSE, so how do we know it was an electron microscope? It had high resolution, and you know that from GCSE. Electron microscopes have higher resolution. You can see more detail. The ability to distinguish between two separate objects as separate, or objects as separate. Oh my gosh. Also, we can see the nucleus. That wouldn't get you a mark. Or any named double membrane bound organelle. Beware of the list rule. E.g., if you say oh, you could see the cell wall. Well, you could see that with an optical microscope as well. So use your double membrane bound organelles. However, you could see the nucleus. So scrap that one I've said about the nucleus, because you could see that with an optical microscope. We'll cross that one out there. What we really want to say is, like, you could see the nuclear envelope with an electron microscope. So you could see the membrane of the nucleus. You could see the nucleolus inside the nucleus. You could see the mitochondria. So really, with the electron microscope, you're going to see the smaller organelles, which but you're not you can't really say, oh, I'll see the nucleus or a cell wall, because those are organelles you can see with a light microscope. And you definitely get a mark for higher resolution. All right, let's look at the next one. State one role of a helper T cell. So what do the T cells do in immunity? The T cells are those ones that bind. Remember, they have like a receptor site that binds to the antigen, or sorry, binds to the protein that's being presented on the antigen presenting cell. So it's those T cells that bind to that, and then they activate either T killer cells or toxic cells, or they activate B cells, and the B cells divide into plasma membrane and memory cells. So really we want to put stimulate B cells, stimulate T killer cells, or stimulate phagocytes. Again, beware of the list rule. Just say one thing they do that you know for sure, because it only asks for one of these. So when it specifically asks for one, if you say two and one of them is wrong, they're going to minus that mark off. Right, 4.4. The rate of translation is increased in T cells activated by antigens. Describe the role of tRNA in translation. That first line we didn't really need. They're just asking you, what does tRNA do in translation? So tRNA is a carrier of amino acids. It's the carrier molecule for amino acids. So you're going to get a mark for saying tRNA carries amino acids or brings amino acids to the ribosome or the mRNA. We then want to say that tRNA, what's the other special thing you know about it? It has an anticodon that is complementary and binds to the codon on mRNA, and that is your 2 marks. I mean, just for extra information, tRNA also holds the amino acids in place while a peptide bond forms between the 2 adjacent amino acids. And then the tRNA leaves and another tRNA comes bringing another amino acid. But it's kind of there as like a scaffolding to hold those amino acids in place until peptide bonds form between them. Otherwise, the amino acids could wander off. Now, should I be writing extra waffle like what I've highlighted here in the red box? I shouldn't really be writing this extra stuff for a 2 marker. However, the list rule doesn't apply here in this situation. So they're not going to, this is just a third piece of information. They're not going to subtract this, and it's correct. Make sure any information you write down is correct science, not incorrect, because here they're not going to penalize me for writing this. And in fact, it may gain me a mark depending on the mark scheme. Okay, 5. A student investigate the effect of changing the surface area on the rate of diffusion of a solution into the center of an agar block. She used the agar colored by an indicator. The indicator is pink above pH 8 and colorless below pH 8. She cut the blocks in different shapes shown. Complete the table to show the surface area and the volume ratio of the 2 shapes. So we want the surface area to volume ratio of the 2 shapes, which means I need to count. They've given us the volume is 64 with each, but let's calculate that surface area. So the first block C is 4 times 4 times 6 is 64. And that would give me that would give me 4 times 4 is 16 times 6 is 96. So 96 centimeters squared. And the volume we already said is they've given it to us, but just to show you how they worked it out, it's 4 times 4 times 4 here. Height times width times depth. Etc. So yeah, so we've got a ratio of 96 to 64. So our surface area to volume ratio is 1.5, and I can do that by going 64 divided by 64 is 1, 96 divided by 64 is 1.5, so I'm getting a ratio of 1.5 to 1. So we can see here with my working out. Lovely jubbly. Right, the second one is slightly more tricky because the diagram looks confusing because it's not really very scale-like. So the surface area of this one is 16 times 2. Now remember that's going to be 32, but there's 2 sides that are going to be like that. There's one on the front here and then one on the back. So that's a total of 64 we've got. And then we've got 2 other lots, well, of 16 times 2. The one on the top and the one on the bottom that's hidden from us is also 16 times 2. So we're going to have a total of 128. And then if you look at the 2 ends, they're 2 times 2. And so we've got 2 of them. So 2 times 2 is 4, and we've got 2 of them, one end at each end there. And so when you add up the surface area of D, it is 136. So it has a bigger surface area than C, but let's check its surface area to volume ratio. 136 to 64. And that equals 2.125. How do we get that? We do 136 by 64 equals 2.125, and 64 by 64 equals 1. So it's 2.125 to 1. And there we go. 2 beautiful marks. Let's move on. Okay, point two here, 5.2. The student put the blocks in an acidic solution. The acidic solution caused the blocks to gradually turn from pink to colorless. She recorded the time taken for the block to turn completely colorless. She repeated this 3 times. So we've got block 1, 2, 3, and 4. And we got the mean time for the block to turn colorless. After collecting the data, the student noticed the shape of block 2, shape C, block 2. This one here I've circled anyway, just because I'm getting myself confused. This one is damaged. Calculate the mean for shape C. So what do we do with anomalies? You know what we do. We take the anomaly out, we remove it, and we kick it in the bin. And so what we want to do is add up the other 3, block 1, block 3, block 4, and divide by 3, and that'll get us our correct answer. Correct answer I was supposed to say there, getting a bit of technical faults. Right, so calculate the mean for shape C. We want to subtract the anomaly, get rid of the 1,200, add up 3,490, 3,540. What's that last one? Is that 3,530? My eyesight is so bad. Add them all together, divide by 3, whack that in this calculator, bing bang bong. What do we get? Hopefully you got the same as me. That's 3,520. And that'll get you a mark. All right, let's look at the next question. Suggest what the student should have done when they saw that shape C, block 2, was damaged. They should have chucked block 2 away for shape C and brought in a new block, or repaired the damaged block. Yeah, returned it to its original shape. That's what they should have done. And that'll get you a mark. Right, let's state 3 variables. Oh my gosh. Um, State 3 variables the student controlled in order to obtain valid results. So what are 3 things they should have controlled? Think about this experiment. Have a look what we're using. We're using an acidic solution. What should you keep the same about the acid? What should you keep the same about the agar? Um, what should you keep the same about the time they were left in the acid? So think of all that. It's really about diffusion. So anything really affecting the rate of diffusion, obviously how long you leave it for, the concentration. So I'm thinking concentration of acid is pretty good. The temperature would affect the rate of diffusion, so probably use a water bath for this one. The type of agar might come in different types. Some might be more porous than others, so diffusion will take place more easily. So there's all sorts of ones. The concentration of the indicator, i.e. if you've used more drops of phenolphthalein coloring one, it's more pink than another, then it might take more acid to go colorless. So you've got all of those types of things that could affect the accuracy. All right, any 2 of those will do. Let's rock. 3 of those, sorry, any 3 of those will gain you 2 marks. Oh, it's late today. Right, 5.5. Describe how gas exchange occurs in single-celled organisms and explain why this method cannot be used by large multicellular organisms. Once again, I've used the highlighter just to remind me of the key facts we're talking about here. And really, this one, what do you remember about single-celled and large multicelled? Single-celled organisms, remember, have a large surface area to volume ratio. This means simple diffusion alone can meet their requirements for glucose, for oxygen, for other sort of metabolites. If you're a large multicellular organism, simple diffusion will not bring the materials into the cells quick enough for them to do all of their different metabolic processes they need to do, like aerobic respiration. The cells deep within the organism, they're not going to receive enough oxygen and glucose by diffusion alone, which means we need mass transport. We need things like ventilation with the lungs. We need a circulatory system with the blood. So having a low surface area to volume ratio, which large organisms do have, means that we need specialized exchange surfaces. So our lungs, of course, can actually absorb a huge amount of oxygen for their actual volume because they've got a really large surface area to volume ratio. The lungs do, and our small intestines as well. So this is why we have specialized surface exchange surfaces like the lungs with the alveoli, like the small intestine and the microvilli and the villi. This is why we have those. And it's also why we require mass transport mechanisms. And it's the same for plants. Plants plants. Plants have the root hair cells giving that large surface area to the roots, but they also have a mass transport system with transpiration, the cohesion, tension, and all of that. So, check this out. Here are the answers that are going to pick you up 3 marks. Any of these will get you sorted. All right, that's the. Okay, question 6. The human papilloma virus contains a double-stranded DNA genome. Which components are found in the human papilloma virus? So all of this is waffle to try and throw you off. It's simply them asking, what do you find in a virus? What does a normal virus contain? Only tick one box. So we've got, does it have a capsid attachment protein? A capsid attachment protein and reverse transcriptase, you know, that enzyme that helps them make DNA from RNA. Does it have a capsule and an attachment protein? Does it have a cell surface membrane and attachment protein? So what we need to think about here is which of these ones is actually correct. Well, let's start at the bottom here. Cell surface membrane and attachment protein. Well, it's not going to have a membrane, is it? It's a virus, and a virus doesn't contain any of the organelles found in cells. A virus is not a cell, it's a tiny particle. In fact, a lot of scientists do not consider them living things because they can't do the metabolic processes of cells, and they don't contain any cellular structures like cytoplasm or cell membranes. So it's definitely not that one. Let's look at the next one, capsule. Where have we heard the word capsule before? Remember that earlier question with the bacteria and the murin in the capsule, the slime layer? Well, that's for a bacteria, not for a virus. So this is definitely an incorrect answer. This can't be something that you would find inside a virus. Um, it sounds an awful lot like capsid, a capsule. So these are two to get that people will get confused if they haven't been doing their revision and making good flashcards and memorizing all those key facts. So a capsid is the outer layer, the protein coat of a virus, and of course they need attachment proteins. That's how they inject their contents into cells. The host cell is via the attachment proteins, but they don't actually contain reverse transcriptase, the enzyme. The virus actually makes enzymes or uses enzymes that are already existing in the host cell. So our actual correct answer is the top one, and that is that you would expect in a virus to find the capsid, the protein coat, and an attachment protein. And that's the answer for 1 mark. All right, we're going to move on here. And so we're going to look at 6.2. So let's go. 6.2. The DNA replication enzymes of a human cell make copies of the human papilloma virus genome. Name 2 enzymes that are involved in replicating the DNA of the human papilloma virus and describe their roles in the replication process. So, what all of this sounds really complicated, but when you break it down with a highlighter, they're just asking you what 2 enzymes are involved in DNA replication, and you guys know that. It's DNA helicase, which breaks the hydrogen bonds and separates the strands of DNA, and we're going to have DNA polymerase, not RNA polymerase. Again, a simple mistake. RNA polymerase is involved in protein synthesis, that's the transcription. It's DNA polymerase. And what's the job of DNA polymerase? It forms the phosphodiester bonds between the 3 nucleotides that have lined up along the template strand to form the new strand. Basically, the DNA polymerase builds the sugar phosphate backbone by forming phosphodiester bonds. And then we're going to get our lovely 3 marks here. So let's crack on. Here we go, 6.3. Human papillomavirus infects cells that are no longer dividing. The human papillomavirus genome contains genes that code for proteins that cause human cells to restart their cell cycles. Human papillomavirus infection can cause cancer. Explain why. Remember, it's basically asking you what is cancer? What's it linked to with the cell cycle? It's linked to mitosis. Cancer is uncontrolled mitosis or uncontrolled cell division. And that's all we have to write for 1 mark here. Okay, let's sing down and check out 6.4. 6.4 Human papillomavirus HPV is transmitted through sexual contact. More than 95% of cervical cancers, which only affect females, are due to HPV infection. HPV infection of other tissues increases the risk of cancer, but it's rare compared with cervical infection. A vaccine is available. Over 80% of the effective at presenting HPV infection. Can't speak. If given before the person has been exposed to HPV, there is evidence of herd immunity when more than 50% of the population have been vaccinated. Evaluate whether 10 to 12-year-old boys should be given the vaccine. So we got some information there to give us some answers. Remember, we're looking at arguments for and arguments against. There's a lot of arguments for here. One of the key things to spot, which I highlighted there, was the herd immunity bit. In order to achieve herd immunity, you need more than 50%. Obviously, roughly 50% are male and female. So if we need more than 50%, like 52, 53%, we're going to have to vaccinate some boys as well as the female population. Otherwise, we can't get more than 50%. So that's a good argument for vaccinating boys. Also, if we vaccinate the boys, it's going to reduce the spread if given to the boys before they get sexually active. So a boy with HPV virus will not be able to pass it to a girl if we vaccinate them at the age of 10 and 12. That's a key age to do the vaccinations before most people are sexually active. So that's a good point to vaccinate them. Then you wouldn't say vaccinate 25-year-old boys because by then they might be sexually active. Also, another element is that it protects the boys from cancers in other tissues, not to do with cervical cancer. But remember, this is rare, so this is only a minor benefit, but it's still a benefit when you're writing an evaluation. So it also reduces the chance of boys developing cancer in other tissues. Now we come to the against. What is the most likely cancer for you to get if you have the HPV virus? And the most high risk of cancer for you to get if you have the HPV virus is cervical cancer. And boys, number one, don't have a cervix, and so therefore they cannot get cervical cancer. So that's a kind of a point against. Is it really worth spending billions of pounds vaccinating all boys in the country when you could be spending that on things that are of greater risk? And we also don't know the side effects in boys. We don't know if there are any rare or side effects from the vaccinations. Okay, nice. So there we go. Most of these were for, just a few against, and let's move on. Okay, let's go. Where are we? Bada bada bing. Question 7. Scientists investigated biodiversity in prokaryote communities found in soil. The scientists took soil samples from fields that had been managed for 20 years with two different farming methods, farming method 1 and 2. So let's go down, blah, blah, blah. If you want, go back, pause the video and read that. I can't be bothered to read all that. 7.1, using the standard deviation data from table 4, describe the differences in the prokaryotic biodiversity found in the soil with these two farming methods. Now it tells us in your answer, give two definitions of species richness and index of diversity. So that's worth 2 out of the 4 marks. And the rest of the marks are going to be commenting on the standard deviations. So essentially, you're looking at basically just define species richness, which is the number of different species um, within a habitat or an area, right? Is the number of different species. And the index of diversity is essentially the species richness, so the number of different species, along with the species evenness, which is the relative abundance of each species. And so index of diversity, of course, is that calculation we do where we take into account abundance and evenness. Anyway, let's go and look at so that's going to get you 2 of the marks. Now let's look at the standard deviation information. All right, let's zoom in. Actually, scrap the zoom in. I'm going to need all this for my working out. Let's take farming method 1, the mean species richness. What can we say about it? Well, it has SD or standard error bars of plus and minus 17. So we're going to go from the mean of 517, add 17 to that, you're going to get 534. Take away 17, you get 500. Is there overlap between farming method 1 and farming method 2? So farming method 2 is a mean of 560 with a plus or minus of 24. So it goes from 560 to 36. Now I'm going to move this just to show you. Obviously, the mean of 560 is going to be a lot bigger, but look at this. There's a gap here. So that goes up to like 584. But there's a gap here between 536 and 534. There is no overlap in the standard deviation between farming method 1 and farming method 2. This means we can 100%, not 100%, 95% say that that difference in the means between farming method 1 and 2 is not down to chance. There's actually something to that, i.e., farming method 2 gives you a better species richness. Well, let's look at the next set of data. We've got the mean index of diversity. Let's check if the results here, when we compare them, are significant. So we've got a mean of 251, it goes down to 240 and up to 0.262 because I've got a standard deviation of 0.011. So you just add that and subtract that from the mean. Let's look at the next mean. The next mean is 230. We've got 0.014 I need to add on. That means it goes from 230 to 0.244. Check this out. I'm not going to bother with the others. There is overlap. As soon as there is overlap between the data, I can't say that farming method 1 or 2 is better at preserving or at getting a mean index of diversity. Okay, so we cannot say that in our answer. It looks like there is no significant difference in the index of diversity between farming method 1 and 2. So species richness, let's go for our 4 marks. We want a definition of species richness. Species richness is the amount of different species in a region, habitat or area. There is a significant difference. I'm going to stick with species richness here and then go to the stats. So there is a significant difference in the species richness between farming method 1 and 2. 2 has the greater species richness, and it is not due to chance. We know that, and I need to point out there is no overlap in the SDs. So I've already done that on the right. I'm not going to rewrite it again, but you would put that in your answer. No overlap in the SDs, standard deviation for farming method 1 and 2 when we were looking at the results for species richness. However, with the index in diversity, we're going to get our mark for the definition here. Index of diversity is a calculation or a measure of the biodiversity based on the species richness and evenness, which is the relative abundance of each species. Now, there is overlap between the standard deviation in the index of diversity. Okay, we've got overlap between the means. And this means there is a 95% chance that any difference in these means or 95% probability that any difference in the means is down to chance. So we can say that the difference is not significant. Okay, that this difference is not significant. Right, let's move on. Hope you got all of that info. Okay, genetic diversity in soil species was traditionally inferred by making observations after growing prokaryotes on agar plates. However, it is estimated that 10% of prokaryotes found in soil will grow if spread on agar plates in the laboratory. In recent years, our knowledge of prokaryotic biodiversity in the soil has increased, suggests why. So why do we know more about the biodiversity of prokaryotes in soil? And the answer is we've got new technologies. We can now sequence the DNA of the bacteria, or we can now sequence the proteome or the amino acid sequence, and therefore we can now identify and analyze more prokaryotes within the community. Now, for the next question, we're being asked to evaluate the balance between conservation and farming for method 1 and 2. So let's have a little look here. Look at the mean total prokaryotic biomasses. You get more prokaryotic biomass with farming method 2. You get more carbon locked up, but you get a lower yield. So those are the points we need to make to get the 2 marks here. So farming method 2 has a greater species richness, as we know. We also have a greater biomass of prokaryotes. So if you use farming method 2, you're going to have a larger population of prokaryotes within the soil. It's also better at locking up carbon. Did you see the mass of carbon up in the data table was greater for farming method 2? And that's going to mean there's a better effect on locking up carbon and reducing the greenhouse effect. So those are good points. However, it did produce a smaller yield of wheat in the farming method 2. So although it might be better for conservation, better for reducing the greenhouse effect, you get a higher species richness, you get a greater biomass, you actually don't produce as much food for like food security and feeding people. Basically, you get a lower yield, which might mean you need to increase the amount of farmland used and things like that. So yeah, it's all a balance. All right, let's zing on. Just 2 questions to go. 8 and 9. 8.1. A student prepared a plant root to observe the cells undergoing mitosis. He put the root in a small bottle of hydrochloric acid in a 40-degree water bath. Why did he put the plant root in acid? So what do you think the hydrochloric acid is there for? It's to break down the cell wall. Well, if you remember when you do your reducing sugar, non-reducing sugar we sometimes add hydrochloric acid. And that's because it can break glycosidic bonds between certain monosaccharides. So actually, it helps hydrolyze the cellulose in the cell wall. We could also say that it's useful for separating two different cells or separation between cell walls. That's the middle lamellae that it breaks. State 2 precautions required when using hydrochloric acid. So nice and easy. Make sure you wear goggles, make sure you wear gloves, lab coat, anything like that would be suitable. Okay, let's go to 8.3. Figure 7 shows the scientists' results. We have, well, what did these scientists? They determined the mean mitotic index at 50 micrometer intervals away from the root tip in 10 young shoot plants. So we have distance from the root tip along the x-axis, and on the y, the mean mitotic index. State the null hypothesis for this investigation. So look at your y and x-axis. We want to basically say there's going to be no significant difference, and then we go to the x-axis, between the distance of the root tip and then the y and the mean mitotic index. So there's no significant difference between 200 micrometers and 300 micrometers, which is what they asked us to do here, because the student wanted to determine whether there was a difference between distance of 200 and a distance of 300. And what we're saying is, with a null, is that there will be no significant difference between the mean mitotic index when the distance from the root tip is 200 micrometers or 300 micrometers. Now, what statistical test would show this? When you're comparing two means, you're going to use the t-test. Awesome. And that'll grab you 2 marks. Let's move on to 8.4 and crack on with the rest of the test. So 8.4, the scientists recorded the measurements from the tissue located at 200 micrometers from the root tip. Geez. The area of the view of the field of view, that means when they're looking through the eyepiece lens, they can see 0.2 millimeters squared. The mean area of one cell was 3,495 micrometers squared. Use this information in figures and figure 7 to calculate the number of cells undergoing mitosis in the location. Assume there are no spaces between the cells, so every single part of that 0.2 millimeter area of view is filled with cells. We need to use figure 7 as well. So I'm going to draw a line up from 200 micrometers and we'll find the mean mitotic index is 0.35. Now we're going to need that. So let's go back down and do our calculation, bearing in mind that the mean mitotic index is 0.35. Now here you need to know the equation for calculating mitotic index. You should know the equation. It's the number of cells undergoing mitosis divided by the total number of cells. And what they're asking for us here is use the information in figure 7 to calculate the number of cells undergoing mitosis. Okay, so we're going to divide we need to times the mitotic index by the total number of cells in the field of view. So 0.35 times the total cells in the field of view will give us the cells going through mitosis. So we need to work out how many cells are in the field of view. Now, bear in mind that 1 millimeter times 1 millimeter, i.e. 1 millimeter squared, is 1,000 micrometers times 1,000 micrometers. So 1 times 10 to the 6 micrometers are in 1 millimeter squared. We've got 0.2 millimeter squared, so we need to times that by 1 times 10 to the 6. And so if we do 0.2 times 1 times 10 to the 6, or times a million, you're going to get that answer. What is 0.2 millimeters in micrometers? Well, it's 200,000 micrometers. 200,000 micrometers squared in the field of view. I can't be bothered to put the little 2 there. Okay, so that means that we have a field of view with 200,000 micrometers squared. We know that one cell makes up 3,495 micrometers squared. So how many cells could you fit in 200,000? Well, you're going to do 200,000 divided by 3,495, and that will give you the total amount of cells that we have altogether. So we're just going to do 0.35. If you imagine these were in a triangle, you'd have cells in mitosis at the top and mitotic index and total number of cells at the bottom. So we're going to times 0.35 times 200,000, which is the field of view, divided by the area of the cells, and that is going to give us our cells that are undergoing mitosis. And the answer is that it's about 20.2, or we can 0.02, we can round that to 20 cells are undergoing mitosis. Oh, I think there's something like 50. If you do 200,000 times 3,495 divided by 3,495, I think you get something like 50 or 57. But that was in my calculator. I just bashed it all in together. So another student compares the mitotic index in the roots of different species. What two things do we need to ensure is kept the same? What are the control variables? Now, whenever they mention validity, that's all about your control variables. So remember, we used hydrochloric acid, so we want the same concentration of hydrochloric acid. Some other control variables will be the same distance from the root tip with both species, the same time we leave them in the acid for both species, and the same age of the root tip for both species. So there's 4 control variables there. You're going to get a maximum of 2 marks. It only asks for 2 considerations, so don't write 4. That was just me showing you the different possibilities. Okay, moving on. We're getting near the end now. We're on question 9. So let's have a go at this. I'm not reading all of that. If you want to read that lovely jubbly information, pause the video now and have a explain how the sucrose transport proteins in leaf cells enable the production of a pressure gradient in the phloem. Basically, this question for 3 marks is asking you explain how we get a pressure gradient in the phloem. Mass transport. Well, that involves the active loading of sucrose into the top of the phloem from the companion cell. Now, you can say this is by active transport through a transport protein or a carrier protein, or you can say it's facilitated diffusion through a channel protein. Either of those are going to get you the marks. Now, you might be asking, well, is it active transport or facilitated? It's actually a combination of both, but in the AQA syllabus, you don't have to know how it exactly works. You can just say active loading of sucrose into the top of the phloem. Now, what does that do? That lowers the water potential because you're adding sugar in there. So the water potential gets lower. Water moves in from the xylem, which is right next door to the phloem, separated by some material called a cambium, which you don't need to know in AQA. So water moves in by osmosis, and this creates a high hydrostatic pressure at the top of the phloem, which is going to force the fluid in the phloem down towards the sink, away from the source. And that's how we get a pressure gradient. Now, we could also talk about why the pressure is low at the bottom, and that is because sucrose is being used in respiration or converted to starch at the sink. Right, let's zing on. 9.2. Potato plants with fewer sucrose transport proteins produce fewer potatoes and accumulate sugars in their leaves. So they're getting less transport to the roots. Why is that? They have less transport proteins, so they have less protein channels or less carrier proteins, which means less sucrose is actively loaded into the phloem. We end up with a smaller water potential, less water moving in by osmosis, so we end up with a smaller pressure gradient. This means less sucrose moves away from the source to the sink, which is the roots in this situation, and so more of it is stored in the leaves. But Okay, 9.3. During exercise, there's a significant increase in glucose uptake by the muscle cells, lines 14 and 15, give 2 reasons why glucose uptake in the muscle cells increases significantly during exercise. Explain your answers. Right, so why do we get increased uptake of glucose in the muscle cells during exercise? Well, we have increased aerobic respiration in order to produce more ATP. You might not know yet because you're doing AS, but ATP is used in muscle contraction. It's actually used for breaking actin-myosin cross bridges, those of you that are studying A-level at the moment. So it's a vital part of muscle contraction. It's also used for the power stroke, the actual contraction of the muscle. Oopsie, cut myself off there. So basically, for the first 3 marks, I would say increased aerobic respiration means more ATP. Increased aerobic respiration to produce more ATP for muscle contraction. That's 2 marks. That increases the concentration gradient because you're decreasing the amount of glucose in the cell, so you're increasing or making a steeper concentration gradient. We're also told in lines 14 and 15 that there are these transporter glucose proteins, channel proteins, that are stored on an inner membrane, and then during exercise they move into the plasma membrane, allowing a greater uptake of glucose in the cells. So we're going to get some extra marks here. We've already got 3 for saying respiration, aerobic respiration, sorry, more ATP used for muscle contraction, greater concentration gradient or steeper concentration gradient of glucose into the cell. And we're then going to get an increased amount of channel proteins for increased facilitated diffusion of glucose or co-transport of glucose. An increased amount of channel proteins are moving into the cell membrane. Now let's look at the last point. 9.4, we're nearly at the end of the test. Which statement correctly describes the structure of both a sucrose transport protein and a glucose transport protein? Let's get rid of incorrect stuff. So secondary structure held together by disulfide bridges, that's nonsense. And the second one, secondary structure is disulfide bridges. Nah, we know it's hydrogen bonds that holds the secondary structure together. Woohoo! The last 2 answers start correctly, so we're not sure which one it is. Then says the tertiary structure allows the protein to be positioned on the outer surface membrane, and then we have the tertiary structure allows the protein to be positioned spanning the whole membrane. Yes, of course, intrinsic protein, we want it to be a channel to allow the glucose or the sucrose through. It needs to go all the way through the membrane. So the final one, that's the answer. And that's the end of the test. Let's get to the marks.
You're still here? It's over.